Thanks for the introduction. I'm Masroor Bayati. I'm doing my PhD at the Medical Biophysics Department at U of D, and I'm also a research assistant at OICR. And today I'm going to talk about the inferring the pan-cancer interactions of prognostic significance of driver mutations and the tumor immune microenvironment. Tumors evolve through the accumulation of somatic mutations. A small fraction of somatic mutations are drivers of cancer since they enable oncogenic properties. During cancer progression, different driver mutations may give rise to different subclones of cancer cells with distinctive pathway activities. But somatic driver mutations are not independent of the tumor microenvironment. Through immune editing, subpopulations harboring different mutations undergo selection pressure of the host immune system. And on the other hand, rapidly dividing and mutating tumor cells can secrete cytokines, for example, that recruit certain suppressing immune cells. All in all, the bidirectional crosstalk of the immune genome and the patient's immune system contributes to the functional and clinical characteristics of tumors. Several major efforts have compiled comprehensive catalogs of cancer driver genes that are characterized by recurrent somatic mutations and copy number alterations in various cancer types. A recently published pan-cancer analysis leveraged 26 such computational methods and developed a consensus list of 299 driver genes. Another pan-cancer analysis characterized almost all tumors in TCGA with respect to their immunogenomic features. By clustering bulk RNA-seq data, they identified 6 pan-cancer immune subtypes that included mixtures of multiple cancer types, suggesting that the tumor microenvironment, actually the tumor immune microenvironment, has important characteristics across different types and sites of cancer. The study made further interesting observations in that the immune subtypes exhibited significantly different survival patterns and also statistically associated with the presence of specific genetic alterations. Previous studies have shown that the amount of immune cells infiltrating the tumor microenvironment are informative for patient prognosis. On the other hand, mutations in several known cancer driver genes are also associated with patient prognosis. However, the interactions of genetic alterations and the tumor immune microenvironment, as well as their association with patient prognosis, remain less explored. To that aim, we set out to perform a pan-cancer analysis using the genomic, transcriptomic, and clinical data provided by TCGA dataset of about 10,000 samples and over 30 cancer types. There are several tools for enumerating the composition of immune cells in bulk tumors. One of such tools, which is widely utilized, is Cybersearch. Actually, Cybersearch is a deconvolution method that models the observed bulk gene expression dataset from tumors as a linear combination of the expression signatures of user-specified cells. The contribution of each cell signature to this linear combination is considered as a proxy of the amount of those cells in the bulk tumor. And in the context of immune microenvironment, they have shown that a signature matrix named LM22 provides a robust enumeration of 22 leukocytes, including population of T cells, naive and memory B cells, plasma cells, natural killer cells, and subsets of myeloid cells. So using transcriptomic data provided by TCGA, we extracted immune features for each sample, which is a relative abundance of 22 leukocytes in the bulk tumors using Cybersearch. The second, we used the mutational status of driver genes in samples by considering non-silent SNVs in consensus 299 cancer driver genes. And third, we actually determined all the 2x2 interactions between mentioned immune and mutation features, all recorded as binary variables. We define each immune mutation interaction as the co-occurrence of a given driver gene mutation with the high or low abundance of a given immune cell type in the tumor. We then use a machine learning strategy of elastic net regression with Cox proportional hazards combined with Monte Carlo cross-validation to actually select prognostic features and their interactions. We iterate over large number of random subset of cancer patients, and in each round, we fit a Cox proportional hazard model with elastic net penalty as a regularization to extract the survival associated features. And when we repeat this process for random subsamples of data, we finally observe that a small group of features are repeatedly selected by this regularization technique, whereas the others have obtained low selection frequency, suggesting that they are less important features overall. Finally, we select the features which have passed the cutoff of 50% frequency, and we'll consider them as candidate prognostic features for the input cohorts. This giant heat map shows the total of 140 prognostic features detected by our model for the various cancer types in TCGA, where you can see cancer types on y-axis and various immune and mutation features on x-axis. First, we found that the prognostic features are quite diverse among cancer types. The first block on the left shows the scores of immune features that are individually prognostic for each cohort. The following blocks correspond to specific driver genes whose mutation alone or in combination with immune features have been identified as prognostic. Among the driver genes, TP53 mutations and its interactions with immune features are the most prevalent among cancer types. KRAS comes as second, IDH1 as third, and so on as we move to the right on this heat map. Specific driver genes are found for different cancer types as expected. For example, GATA3 and its interactions found only in breast cancer, IDH1 interactions only in lower grade glioma, and KRAS interactions are found in pancreatic cancer and lung cancer specifically. We systematically evaluated the risk stratification power of the candidate features using different sets of input features to our models. First, as the control feature set, we consider a basic set of clinical features, including patient age, patient sex, tumor grade, and tumor stage. The second feature set includes the individual immune features without interacting with any driver gene. The third feature set is the immune and mutation and their interactions. And the fourth feature set is the combination of baseline clinical features and the candidate features of immune and driver interactions. And each box plot represents the distribution of models' concordance index or C index in the cross-validation experiments. And actually, the C index is a measure of models' ability to stratify patients based on their survival outcomes. As you can see on the left, for liver cancer cohort, there is a consistent improvement in models' performance, starting from baseline clinical features in red towards the more complex models, and finally in purple. This trend suggests that immune and mutation features and their interactions are not only better prognostic features or predictors compared to the conventional clinical features, but also their combination improves models' performance, which suggests that the prognostic information within these features are complementary. The same results are obtained for several other cancer types, such as breast carcinoma and lung adenocarcinoma, as you can see on the right. Now we will look into the case of liver cancer cohort in more detail. 5 prognostic features are identified for this cohort. They are the interactions of TP53 mutation with 3 immune features, namely regulatory T cells, CD4 memory T cells, and monocytes. And the other 2 features are M0 and M2 macrophages, which are individually identified as prognostic. Next, the single Cox proportional hazard model was fitted to the whole training data with these 5 features as predictors, and then the risk coefficients was used to measure an overall risk score for each sample, which is named also prognostic index. And stratification of patients in training data into high risk and low risk groups using the risk score will result in the Kaplan-Meier plot on the top right, which shows a successful overall risk stratification. Then, as an independent external validation of these results, we analyzed the liver cancer cohort in the ICGC. And interestingly, as you can see on the bottom right, using the same risk score designed based on the training data, the patients are again stratified significantly in the external cohort. And to explore the functional associations, we performed differential gene expression analysis on the high-risk and low-risk tumors in the TCGA liver cancer cohort. And among 3,500 differentially expressed genes between high-risk and low-risk groups, we can see the evidence of deregulation of multiple genes, actually the black dots on the left, that have actually established implications in tumorigenesis. We have also utilized active pathway tool, which extracts the pronounced cellular pathways by analyzing the scores of differentially expressed genes. And the results is the enrichment map on the right, which visualizes 45 differentially expressed pathways. And the main subnetwork here represents cell cycle regulation as well as TP53 regulation. This is quite interesting because we have started defining the high-risk group solely based on genome-level alteration of TP53. And now we see the implication in the transcriptome level. So these downstream analyses demonstrate that the molecular features highlighted by the machine learning model we used here actually provided potentially interpretable biological mechanisms. And our future directions are validating the results in other independent cohorts, incorporation of other omics data such as DNA methylation or copy number alterations to find novel prognostic interactions and potentially improve the ML methodology. As the current model is only able to capture linear associations in the data, but the reality is, of course, much more complex. And with that, I want to thank my supervisory committee and my friends in Raymond Lab, and thank you for attending this presentation. I'll be happy to take your questions.
Right. Thank you for the nice talk. We have time for questions. I think there is one question already. So this is from Musalula Sinkala from University of Cape Town. The question is, how superior is your complex mode to a simple curve fitting model? How do you know that your method works well without ground truth data?
Hi, do you hear me?
Yes.
That's a great question. One way of validating this is to going beyond the training data set and validating the results in the external data set, as I just tried to do so with the ICGC liver cancer data set. This is one way, and the other way is random subsampling training procedure that we are taking. All these together, just with these methods, we are trying to make sure that we are not overfitting, and I think that could address that question if I've taken that question correctly.
Okay. So I have a question. I was curious, does your method apply to non-cancerous data, like other types of phenotypes?
That's a very nice comment. We haven't looked into that, but it could be very interesting to see is there any interesting association between these agents, immune features and mutation features, and then characterizing normal samples. There are a lot of data set out there we haven't looked into, but I will take it as a further step in my project. Thanks.
All right. Thank you, Masroor, for the nice talk. And thanks to all the speakers for this very interesting session.